Good afternoon, everyone. First of all, thank you for giving me the opportunity to share with you today the results of this work about the implication of the immune microenvironment in CLL clinical progression. I have nothing to disclose. One of the most outstanding questions in CLL is why some patients are going to progress while others are going to have an indolent disease. In this sense, several studies have pointed out that the acquisition of molecular alterations at the time of progression, but also at stable follow-up in non-progressing patients, is only observed in a small fraction of patients, suggesting that CLL progression is not mainly driven by clonal evolution. But CLL cells, as any other tumoral cells, are able to evade immune antitumoral responses. So, is actually the immunosuppressed microenvironment in CLL leading to its clinical progression? To address this question, we perform genetic and immunological analysis using paired samples at 2 time points from 2 different groups of patients. From progressing patients, using samples at diagnosis and at clinical progression before receiving any treatment, and from non-progressing patients, with samples at diagnosis and at long-term follow-up. Firstly, to determine whether molecular changes could potentially be responsible for clinical progression in our cohort of patients, we perform whole exome sequencing in pair isolated CLL cells at 2 time points at diagnosis and progressions to study changes in the cancer cell fraction between the 2 time points. And what did we find? We found that although CLL driver genes were present in the 75% of our patients, only half of them exhibited significant changes in the cancer cell fraction, affecting these also a small proportion of the single nucleotide variants detected per patient. While the other 50% of patients that have also progressed, saw a clear genetic stability throughout the course of the disease that cannot explain why they are progressing. But what happened with the immune microenvironment, especially with T cells? Are they going to change? Are they going to evolve? By immunophenotyping of paired peripheral blood mononuclear cells at the 2 time points from progressing and non-progressing patients, we found that CD8 T-cells at the time of progression were enriched in effector memory subsets expressing PD-1, and also that PD-1 was highly co-expressed with other inhibitory receptors, suggesting a severe exhaustion, and that only happened in progressing patients. By further analysis of exhaustion features, we saw that these CD8 T-cells had a terminally exhausted phenotype. But T-cells not only phenotypically but also transcriptionally evolve throughout the course of the disease. RNA sequencing results show that T cells at progression are clearly distinct from T cells at diagnosis in terms of their gene expression, affecting these especially 2 metabolic genes. So now the question will be, what triggers T cell exhaustion in CLL? To cover that, we perform co-cultures, and we co-culture T cells from CLL patients with increasing ratios of leukemic B cells in order to mimic progression in vitro, or with healthy donor B cells as controls. Using this approach, we saw that PD-1 was highly induced in CD8 T cells in the presence of leukemic cells, and when we perform those co-cultures using transwells, we also see that inducement in PD-1, suggesting that this is a mechanism dependent on soluble factors. And which soluble factors might be implicated? We identify interleukin-10 as one of them, as higher levels of this cytokine was found at progression in plasma samples, and when we culture primary samples from CLL patients at diagnosis and progression, we also saw a higher capacity at progression for the production of this cytokine. So to conclude, CLL progression is characterized by limited and non-recurrent genetic changes, while is mainly accompanied by increased terminally exhaustion in CD8 T cells. This exhaustion, especially the expression of PD-1, is induced by soluble factors secreted by malignant B cells, including interleukin-10. We have more data to share, so if you are interested in this topic, please visit poster 2096, and thank you so much for your attention.